Welcome back to another fun-filled lecture. This is Metabolic Pathways, Chapter 6.3. So, essentially, what is metabolism? We know that metabolism is the series of biochemical reactions that happens within the body. Metabolic pathways is going to be essentially a series of linked reactions that begin with a particular reactant and end with a final product or final production. Uh, so as you can see here, I have the examples as the reaction going from A to B to C. So A is going to be the reactant or the thing that is initially starting out with. And then B is going to be the enzyme or the intermediate step, and C is going to be the product, the thing that we wind up with at the end of the reaction. So I want you to think about for example, we have maltose, or I guess an example we could use would be lactose. We'll use lactose, which will be represented by A, and then B we'll say is lactase, and C is the final product. So lactose is broken down by lactase into an intermediate step before it's broken down into its sugars that the body uses for different processes. Um, just kind of an example. So each step in the chemical reaction is catalyzed or sped up by an enzyme, and that's important to know. If you remember from our previous section, what did we say an enzyme was? Exactly, it's a type of protein. Enzyme is a type of protein that can be used for chemical reactions. Actually, they help these metabolic pathways. It speeds them up. That's important to know. So, an enzyme is a molecule that speeds up a chemical reaction without itself being affected by the reaction, and that's also important. So, the enzyme, like I said, it speeds up the reaction. It lowers the energy of activation or the energy needed for the reaction to occur. But it itself is not affected, and we'll see that later on. So ribozymes are made up of proteins. Some enzymes are made up of RNA called ribozymes. They're involved in RNA synthesis, and we know that RNA synthesis is responsible for the production of proteins. Generally, just knowing this definition is important. Nothing really else to say here. Um, So, like I said previously, metabolic pathways begin with a particular reactant. So, if you can remember in um, this example here, I give A being the reactant, so it's what we're beginning with, and C is what we're ending up with. So, um, if you remember chemistry, you remember that your reactants is over here on your left side, and then you have some magic, is what my chemistry teacher used to say. Some magic that happens in the middle, and then you wind up with your final product. So it's essentially just imagine you're combining a bunch of ingredients, then you add in like an enzyme, or you add in this one thing, and it breaks it down and purifies it into your final product. Um, it's another way to look at it. But just some definitions again. Probably pretty important for examination purposes. People love to test on definitions, but substrates are usually the things that we begin with. It's typically our reactants, the thing on the left hand side, and they're converted into products. Again, the A to be the C. The C is the product that we wind up with. So if we don't have enzymes, we can also use heat. Heat is another thing. So sorry about that. Um, but again, getting back, uh, like I previously said, very rarely do molecules react with one another unless an enzyme is added. Um, So if we don't have an enzyme, we can add heat, and we know that heat is going to cause the molecules to collide and cause them to um, uh, cause them to react with one another. So if you're thinking about, for example, boiling water, you heat the water up, and as you heat the water up, it's going to break down those hydrogen bonds, and it's going to cause the liquid to go into the form of a gas um, when evaporation is occurring. So, obviously, we don't have an enzyme for water during that process. So, what we're essentially doing is just adding heat to break that down. So, energy of activation, this is important. This is the energy that must be added to the molecules to cause them to react with one another. So, like I said earlier, they lower the amount of energy required for the reactants to react. So, normally, um, uh, let's say that for let's say that I don't know, I'm going to make this up. Let's say that typically this reaction happens at uh, 500 joules, but we add an enzyme in and it happens at 250 joules. I don't even know if that's the right terminology, but essentially it works for our process. You see that adding the enzyme is essentially causing it to lower the amount of energy needed for that reaction to occur. But I just want to stress that the addition of the enzyme does not change the end result of the reaction. You're still going to have the same product at the end. It's just going to do it in a quicker amount of time. Um, it's kind of hard to conceptualize, but it doesn't matter how it doesn't the product itself does not change. It's just the time it takes to get that product. Um, so by lowering the energy of activation, by adding an enzyme, remember, an enzyme speeds the rate of the reaction, so it lowers the energy of activation. The enzyme increases the rate of the reaction. I want to say that again. The addition of an enzyme lowers the energy of activation, or the amount of energy for that is needed for the reaction to occur. Therefore, we increase the rate of that reaction. And remember, what did we say? It doesn't change the end product. And remember, the enzyme is not affected. The enzyme itself is not affected. I give an example here that your book uses. So sugar in your kitchen cupboard will break down into CO2, or carbon dioxide, and water. This is since carbon dioxide and water have much less free energy than the reactant sugar. So sugar in its natural state has more um, free energy because it's able to break down into something simpler rather than carbon dioxide and water. And that's all that simply means. However, the rate of the reaction is so slow that we typically never see it. So if you just leave sugar laying around, eventually it'll break down into water and CO2. But with the addition of an enzyme like amylase, right? Whenever we ingest sugar in our body is able to utilize it at a much quicker rate rather than just waiting around for it to break down to CO2 and water. So amylase is an enzyme produced by the salivary glands. It's also produced by the pancreas. You don't need to know that, but for you don't need to know that for examination purposes or quiz purposes. Essentially, I just wanted you to know that it is an enzyme that does exist that does break down sugar. So that enzyme speeds up the rate at which sugar is broken down. But the enzyme amylase isn't affected by this reaction. And guess what? We still have CO2 and water. It just changes the rate at which it occurs, how fast or slow it will do that. You can see over here on the right hand side of the screen, we have, like we said, the energy of activation. Um, so no matter what, it's going to get to from a reactant to a product. So sugar is going to break down into CO2 and water no matter what, no matter the path. We'll say that the purple line is without amylase, and we'll say the dotted line is with amylase. So you can see here that the addition of amylase is obviously going to lower the energy of activation, the energy needed, but the end result's still the same. It just happens at a quicker speed. So we know about enzymes, we know what they are, we know what they do, but how do they function? So um, I remember learning this in school. It was very complex. At least I thought it was at the time, but I found a really nice picture to kind of illustrate this. So, enzymes contain an active site, and we can see here. The active site is simply just where the enzyme combines with the substrate. So, we'll say this is amylase, right? And we'll say this is sugar. Well, this site right here is where the substrate is going to combine with, well, for our purposes, sugar. This is where sugar is going to act with amylase. And once it forms this enzyme substrate complex, the reaction is going to occur. So we say here, it is here that the enzyme and the substrate fit together like a key fits a lock. So there's two different types of models, the lock and key model, and there's one called the induced fit model. I think that your book talks more highly about the induced fit model, but I just want you to know that both of these exist. The induced fit model is simply the active site undergoes a slight conformational change to accommodate the substrate. So um, essentially, it's just kind of making it's kind of changing its shape, therefore, it can bind more substrate. The induced fit model, so like we previously mentioned, the active site undergoes a slight conformational change to accommodate the substrate. So the change in the shape is what causes the reaction to occur. So after the substrate, so after sugar and amylase combined, and once we have the production of carbon dioxide and water, the active site of amylase would return to its original shape. So over here, we have the active site. We'll say this again is amylase. This is sugar. Once the sugar combines with amylase, we have this um, amylase sugar complex. Once we have the production of carbon dioxide and water, this would undergo a conformational change and would kind of release um, the sugar molecule. Only a small amount of enzyme is needed in a cell because enzymes are not used by the reactants. So other function of enzymes, we have the digestion of proteins. There's specifically two different types. There's pepsinogen, which is present within the stomach. Pepsinogen gets cleaved into pepsin. You guys don't need to know that. Just kind of giving you an example. And there's also something called trypsin. Your book mentions trypsin. Trypsin digests proteins by breaking peptide bonds. Do you remember peptide bonds, what they do? Responsible for protein, for the binding of different proteins. So the active site of trypsin contains three amino acids. Remember, amino acids are the building blocks of proteins um, that actually interact with the members of peptide bonds. So this trypsin is responsible for interactions with uh, the different amino acids of the protein. And this illustrates the formation of an enzyme substrate complex. That's very important for speeding up the reaction. So essentially, all I wanted you to really know from this slide is that enzymes do have other specific functions um, rather than speeding up a reaction. So they can be responsible for the breakdown of peptide bonds. Enzymatic naming, all this slide is represented as anytime you see any enzyme, it usually ends with ACE, A-S-E. So what breaks down lipids? Lipase. What breaks down urea? Urease. What breaks down maltose? Maltase. What breaks down, let's say, lactose? What would you think? Well, probably lactase. This is going to be probably where the majority of test questions come from. There'll probably be one about, and I haven't written the quiz, but I would imagine if I was looking at the question bank, it would mention something about the induced fit model, probably about the different properties of the enzymes, how they they remain the same, they don't change, they're not used up, how it doesn't alter the it doesn't really alter um, the product. You still get the same result. Lowers the energy of activation. That'll for sure be on there. What else? Um, Probably the induced fit model. I think I mentioned that. Um, so, okay, we'll move on. Factors affecting enzymatic speeds. So, uh, substrate concentration, temperature, pH, enzyme activation, enzyme inhibition. We'll go over these individually. So, depending on the amount of substrate that is available, will depend on the enzyme activity. So here I have the more collision between molecules equals increased speed of reaction. So as more substrate that is around is present, uh, the more is able to fill the active site. So the more sugar that is that is around, the more activity amylase is going to have, because the more sugar is going to be able to bind to the active site of amylase. Therefore, we'll have more product per unit time. Does it make sense? If you've got more substrate or more reactants, you're going to have more of a product. And that's just going to be due to the fact that you're going to have more active sites that are filled. Essentially, it's just think about this. Um, the more you have of something, the more you can make of something. And the more enzyme activity is going to be needed in order to make that something. Um, Temperature is another thing. Remember earlier we said if we didn't have an enzyme, what could we use? Well, we could use temperature or heat. But temperature, in the presence of temperature, you're going to increase the enzymatic activity. And that's essentially just the same thing that we said previously. The more temperature that's present, the more collisions that are going to occur between the enzyme and the substrate. But also you want to worry about something called denaturation, and this is actually something that can happen. You can have brain denaturation. The more heat that is present, so if someone's running a temperature of 105 and 106, they worry, if it's prolonged, they worry about brain denaturation. So what happens is this temperature alters the conformation of the proteins. Therefore, the proteins or the enzymes aren't able to bind to the substrate. So you can actually have a loss of activity of the brain because you have a structural change. So I have here, if the temperature rises above a certain point, the activity level generally levels out and then rapidly declines due to denaturation. And we have denaturation. What is that? It's a loss of biological activity. Um, denaturation, whichever. Remember that enzymes are proteins. Temperature changes the shape of the protein, primarily secondary. What's secondary? What shape did we say? Do you guys remember? It's alpha helices and beta pleated sheets, and tertiary is three-dimensional. Good. As the shape changes, it prevents from binding efficiently to its substrate. So again, temperature is good because temperature increases the speed of which the enzyme can work and will increase the speed of the reaction. We know that, but only to a certain degree, right? Because once you have too much temperature, you can have denaturation. And what happens with denaturation, you have loss of biological activity due to a conformational change, and the conformational change prevents the enzyme from binding the substrate effectively. And then pH. pH. So enzymes require a certain pH in order to maintain their normal configuration. So this would be like pepsin. Pepsin, which is usually present in the stomach, maintains a pH of around 2, whereas trypsin, which is present in the small intestine, digestive tract, is more alkaline with a pH of around 8. So in order for these proteins or these enzymes to work efficiently, they have to maintain a certain pH. If you expose them to a different pH, it's more likely going to wind up with this same different interactions between these peptide bonds, which is going to cause a conformational change. This conformational change is going to change the active site, and therefore they're not going to be able to bind substrate as effectively. Enzyme activation. Not all enzymes are needed by cells all the time. So we have genes that are present that will upregulate or downregulate, um, just depending on the need. So there's something called alpha 1 antitrypsin deficiency. And alpha 1 antitrypsin, trypsin is a protein that is produced, and this is a protein that is responsible for digestion or of something called elastase, and elastase is responsible for breaking down neutrophils. So these people are they develop something called COPD at a very young age, and that's just due to the fact that their body's consistently breaking down the elastin because they don't have elastase present. So this would be an example of the lack of a gene where they would actually need it. Um, but again, not really needed for examination purposes, just in a medical example. Um, Activation of enzymes occurs through a variety of ways. This is probably more important. So, in order to affect the speed of a reaction, we can have activation or deactivation of enzymes. There's something called a kinase, and a kinase is responsible for adding a phosphate group. And this will continue to show up later on for those of you who are interested in the biological sciences. You'll hear this until you're blue in the face: kinase and phosphatase. Phosphatase sounds like it makes sense. Phosphatase means removing of a phosphate group, whereas a kinase is actually an addition of a phosphate group. But I just want you to realize that either one of these can either turn on or turn off a reaction. Usually, a kinase, whenever it phosphorylates something, it usually turns it on. And a phosphatase, I found out, that usually deactivates it, but that's not always necessarily true. But for your purposes, I just want you to know that either way, the addition or the removal of a phosphate group leads to the activation of an enzyme. Other forms of activation include cleavage, or cleaving a protein, removal of a protein. So remember the protein trypsin? Well, it actually exists in its inactive form, something called a zymogen, right? Zymogen. Again, you don't need to know this, but it's just an example of cleavage. So um, trypsin exists as a zymogen in its inactive form, trypsinogen. So trypsinogen is cleaved, a peptide bond is cleaved off of trypsinogen to become trypsin, which activates it. And trypsin is responsible for um, the digestion of proteins present within the small intestine. It helps with absorption through it. You guys will learn about that in anatomy and physiology if you decide to take it. Other ways of enzyme activation is by associating with another protein or cofactor, and we will explain what cofactors are here in a minute. So, what's important on this slide, I would say that knowing that kinases or phosphatases, knowing that they can activate enzymes, knowing that cleavage or cleaving a protein, and also associating with a cofactor, all those are three different ways that enzymes can become activated. Enzyme inhibition. So, what happens whenever an enzyme is inhibited? It means it doesn't work, it doesn't react in the reaction. The activity of every enzyme in a cell is regulated by feedback inhibition. So, what is feedback inhibition? Remember how we said that we have A as the reactants, B as the middle guy, like the intermediate step, where the enzyme's actually functioning, and C is the products. So, when this reaction is occurring, right, you're having this reaction, you're going from A to B to C. Once you have enough product, we can actually turn A off and say, hey, we don't need you anymore, man. Start slowing down on producing this product. This is our body's way to try to maintain homeostasis. Do you guys remember what homeostasis is? It's a characteristic of life, right? And it's also a um, maintaining an internal environment, right? It's our body's way of trying to regulate things. So feedback inhibition is as more products are used up, inhibition is, I'm sorry, when there's plenty of product, it binds up all the active sites of the enzyme, preventing the substrate from binding. So essentially, it's just slowing down the reaction. And it occurs due to the fact that the substrate's unable to bind the active site of an enzyme. So essentially, all the active sites are filled up of this enzyme, which is slowing down the substrate from binding. So this is the way I was always taught: A to B to C. You've got enough C, right? We've got enough products. All the active sites are filled up. Therefore, the substrate or the reactants A can't bind. And I have an example here. The feedback inhibition, you have enzyme 1, right? We're going through, we're producing that intermediate step, like we said. And then you have enzyme 2 going to the end step. Well, whoa, what's happening? We have inhibition of this whole process occurring due to the end product. It's coming back and it's inhibiting this enzyme from binding. So we said that cofactors are another way that enzymes can become activated. But what is a cofactor? Cofactors generally are just mean an inorganic ion. You guys remember what inorganic means? Something other besides carbon or hydrogen, right? So what are examples? Copper, zinc, or iron. But these cofactors may need to be present in order for the enzyme to work properly. So, some of you guys, if you guys ever take biochemistry or just other different physiology classes, you'll learn about cofactors. And the reason that cofactors are important is because they're needed for these metabolic pathways to happen. So, for example, vitamin B12 is an example of, I believe it's an example of a cofactor. Um, or a it's either an example of a cofactor or a coenzyme. But anyways, these are necessary. These are important due to the fact that metabolic pathways can't necessarily happen if we don't have them present. So there's something called vitamin B12 deficiency, and it can cause over time a syndrome called um, it can cause a syndrome over time called subacute combined degeneration of the cord. So what happens is vitamin B12 becomes deficient. Therefore, they're unable to form the myelin sheath over the nerve cells. So what happens is they have demyelination, or the nerves aren't necessarily able to conduct as well, and they begin developing like dizziness. They develop dementia over time, paralysis, paresis, or just like a inability to sense touch. It's a really crazy phenomenon, and what can happen over time is you can give them B12 back, and they can begin regenerating that myelin sheath present around the nerve cell. So it's actually really cool. Don't need to know it. Again, I'm telling you a bunch of stuff you don't need to know, but I just want you to realize that um, vitamins are actually important. So what is important for you is to realize that cofactors and coenzymes are responsible for activation of enzymes. And coenzymes are cofactors that participate in the reaction, may accept or contribute atoms to the reaction. So examples here are NAD, FAD, NADP. I have here what they are made of: nicotinamide adenine dinucleotide. So you can just break it down. Adenine is a nitrogenous base, and we know that dinucleotide just means two nucleotides combined. Um, uh, So you don't really need to know those names specifically. We usually refer to them as NAD plus, FAD. They're essentially just cofactors that if you're in the biology department, you'll get tired of hearing about. So, but each of which plays a role in either cellular respiration or photosynthesis. So cofactors and coenzymes are important. And you can see here on the right hand side of the screen, we've got a cofactor or a coenzyme. It's binding, or it's necessary for the enzyme to become active. The enzyme becomes active, and we have the substrate about to interact with this enzyme. So without it, without the cofactor or coenzyme, this whole process wouldn't even occur. Then here I talk about vitamins. Vitamins are small organic molecules that require trace amounts in our diets and in the diets of other animals for the synthesis of coenzymes that affect health and fitness. So I talk about here kind of just what I previously mentioned. The deficiency of any of these vitamins results in a lack of coenzyme and therefore a lack of enzymatic actions. In humans, these may result in vitamin deficiency symptoms. So your book mentions something called pellagra. And pellagra is just another vitamin deficiency. You don't need to know it. They developed severe dermatitis, dementia, and diarrhea. It's called the 3Ds, and it's just due to a niacin deficiency, vitamin B3. Don't typically see that here. You'll see that in areas that are kind of developing in some of the lower socioeconomic classes. But again, I just included it. Um, Really, I would say if there's main points to know throughout this lecture, I would just know that here's an example of pellagra that I've included. If there's important things to know in this slide or in this PowerPoint, I would know these are important: the substrate concentration, temperature, pH, enzyme activation, enzyme inhibition. Know what cofactors and coenzymes are. Know why they're important. Um, know the facts about enzymes, such as um, they are not consumed or affected by the reaction. Know the fact that that they know the fact that they don't change the end result of the reaction. The energy is less than the products. They lower the energy of activation, therefore it increases the rate of reaction. Um, know the induced fit model, how there's a conformational change that allows the substrate to bind to the active site. Um, again, I put 2 slides in here about the induced fit model. Um, so, if you guys have any questions, shoot me an email. Again, the book is a very good resource. This lecture is just kind of covering some processes that are really too complex to go into. We're trying to give you the guys the foundation of it without going too deep into it, as this is an introductory course. So, again, if you have any questions, shoot me an email. I would be happy to answer them. Um, thank you, guys.